Okay, welcome everyone. It's nice to see that many people here, although it's a little headlights in my eyes, but I can make you out. So today, it's not really a debate, but we're going to give two different sides, sort of, me and Dr. Alan Schindel. I'm going to talk on the basic science side of PDE5 inhibitors. So as we go forward, I have nothing with the ACCME essentials and standards to report. Everyone involved in the planning and presentation of this lecture has no relevant commercial relationship or conflicts, and there is no commercial support for this lecture. I always put that slide up because as a basic scientist, I actually teach to a lot of medical students, and this always stuck with me because I think especially in today's world with the coming out of the pandemic and everybody's afraid to talk to each other. So it's an old slide that's been around, but I think it still carries a lot of weight. So when we talk about what we'll be talking here is mechanisms of PDE5 inhibitors. So just to give a real brief background, here you have a cross-section to the penis. You see the red arteries coming through. They need to carry the blood in. The cavernosal smooth muscle, that pink area surrounding the little tiny dots, that needs to enlarge. And if you look on the right side of the slide, you see an enlarged artery bringing more blood in. The cavernosal smooth muscle is relaxing and filling with blood, and the penis can become erect. If we go a little deeper, we don't have time to go too deep here, but what you can see in this slide is up top you have cholinergic nerves and adrenergic nerves, where cholinergic nerves are releasing acetylcholine, which is going to signal and cause endothelial cell to activate eNOS and release some nitric oxide. And down the bottom you have a nerve, the primary nerve, a cavernous nerve, which is going to release nitric oxide. Then the nitric oxide, what we're going to talk about today, is that activates an enzyme called guanylyl cyclase, which then produces cyclic GMP, and that cyclic GMP in that pink circle above there can then lead to activating a lot of proteins, and those proteins that are activated in enzymes are what's going to lead to smooth muscle relaxation. But what we're going to look at today is, you look all the way down on the bottom of the slide, is that PDE5 inhibitors can prevent PDE5 from breaking down cyclic GMP. So this just zooms in on that one area. So nitric oxide activating guanylyl cyclase, cyclic GMP produced, activating a number of cyclic GMP dependent kinases, and those proteins doing a bunch of things which is going to lead to smooth muscle relaxation, including sequestration of intracellular calcium. This I put in as kind of a funny slide in that my first postdoc way back in 1993, I was in a PDE4 program at Wyeth Ayres Pharmaceutical, and that was a DEA ecephalose column that I was collecting PDE4 and dumping the rest of the PDEs down the drain. And lo and behold, some years later, I'm looking at smooth muscle and contraction, and the key player appeared to be PDE5. Phosphodiesterase type 5, we have 3 isoforms. Exists as a homodimer, so 2 equal subunits. Total weight is 200 kilodaltons. It's localized to chromosome 4q26, and it hydrolyzes only cyclic GMP, which is one of the key points of the PDE5 inhibitors. It was thought that they only worked in the penis, but that turned out not to be exactly true. It'll work wherever there's sort of smooth muscle around. And here you can see, when you look at the one of the PDE5s, you can see that there's 3 components. There's the N-terminal fold, which is circled down there on the right. And then you have the linker region, which kind of connects to the L-terminal helix. So there's 3 isoforms of PDE5, PDE5A1, 2, and 3. In their expression, PDE5A1 is expressed in all tissues, A2 is expressed in all tissues, but PDE5A3 is expressed predominantly in tissues with substantial amounts of smooth muscle. So differences between isoforms in the 5' end of the mRNA and the corresponding N-terminal region of the protein are very important. And here you can see, if we look at the mRNA level, you see A1, A2, and A3, and where that changes, it's in the first exon. And at the very bottom, you can see that those little triangles are showing where there's an ATG, where protein synthesis takes place. So 2 and 3 region is the same, and up top, if you look at the PDE5, you see that cyclic GMP binds to GAP-A, which then starts to activate the PDE5. You have dimerization through GAP-B, and there you can see PDE5i, which we're going to use throughout the talk, doing its thing. Here, if you look at the molecular domains, again, more zoomed in, you see the violet region to the left is the N-terminal domain, the blue region, the C-terminal domain, the linker region, and there you see vardenafil, which is stuck in the active site of the enzyme. And you see a magnesium and a zinc because PDE5s are actually metallo enzymes, and they need those two to work. And here's just if you really want to get into it, you can zoom down to the catalytic domain. It's conserved across isoforms, 10 to 15 to 10 to 20 angstroms, a volume of 330 angstroms, and a narrow opening. In the active site, you have these regions called the core pocket, the hydrophobic pocket, the lid region, and the metal binding site that we just talked about. So here's the big 4 PDE5 inhibitors. So the big 3 that came out first was sildenafil, vardenafil, and tadalafil. And you can see that sildenafil and vardenafil look very similar. They both have 4 ring structures, but if you look closely, there's minor differences. And those minor differences are enough to have a huge effect on the IC50 of vardenafil compared to sildenafil. So at the basic science level, there are differences that can have effects that are important. And tadalafil, you see, has kind of a different structure, and tadalafil is sort of protected from being taken out and passed out of the system, so therefore it has a longer half-life, and that's important for dosing and men that want to have a longer time period. Avanafil, those 4, as far as I know, are the ones that are approved for actual use in the US. Avanafil is supposed to have some isoform selectivity as one of its advantages, and also it has an IC50 not that much higher than vardenafil. If you look at concentration-dependent effects, you can see the square boxes all the way over to the x-axis at 0.0001 nanograms, which is actually 0.1 femtograms, you start to have an effect in relaxation with vardenafil, but you see that tadalafil and sildenafil don't have as much of an effect and comes on later on. This just shows that, again, small differences in these molecules can have an effect. So here's sildenafil compared to methyl sildenafil. Down the bottom, you see those 2 lines that are clearly different changes in the IC50, and up top you see vardenafil compared to dimethyl vardenafil. These last 3 structures I just put up for completeness. The one on the left, eudanafil, has a rapid onset, long duration of action. Microdenafil, orally disintegrating film, and lodenafil actually is given as lodenafil carbonate, and then actually breaks down to the 2 molecules of lodenafil. They actually worked on something that was pretty neat, an article where they are trying to detect PDE5s in various supplements that are offered over the internet. And basically, I won't go through it all, but they prepare a matrix consisting of a powdered dietary supplement, and then they spike that with different PDE5s, and they were very successful in using that. And you can see that when they have a table there comparing PDE5 inhibitor potencies, it matches up very well what they did on the left to what was in the literature on the right. So the second part of my talk is basically looking at touching, because we don't have a lot of time, on other areas that are important. So you can see on the left, approved applications. We talked about ED, but also LUTs, PAH, obesity, Then if you look on the right, there's ones not yet approved, including cancer, cardiovascular disease, neural disease, and COVID-19. And it's all because of PDE5 preventing the breakdown of cyclic GMP, as you see down the bottom, and cyclic GMP doing all that. So no, I would say all PDE5Is are not the same. Each PDE5 has different pharmacokinetics and pharmacodynamics. The degree to which each PDE5 inhibits other PDE5s are different. For example, sildenafil inhibits PDE6, which is present in retina, causes temporary visual effects. The half-life of PDE5s differ dramatically, especially with tadalafil, which has a 17.5-hour half-life compared to 4 for the other two. And food, especially fatty food, affects the pharmacokinetic properties of sildenafil, vardenafil, and tadalafil. So there was a study done, which I thought was kind of interesting, which PDE5I is most effective in the treatment of ED in men with spinal cord injury, which, as you can see there, is pretty prevalent. And there were no head-to-head comparisons. So what they did was they addressed that by using a network meta-analysis, which is a technique for comparing 3 or more interventions simultaneously in a single analysis by combining both direct studies and indirect evidence across the network studies. And they basically sifted through the literature and came up with 10 studies. And you can see, I just put this slide on, when you can see at the studies that are done at the human clinical level, you can see, especially if you look at the diagnostic tools, they're quite different between them. So that's why that study was important, because we don't really have like 3 or 4 clean studies comparing one to another, or all 3 of them. But using that technique, what were they able to find is that the IRR, which is kind of similar to like an odds ratio, was actually positive, meaning it was a positive effect. And tadalafil had the highest effect. And sucra, which is the surface under the cumulative ranking curve, was also higher, so you would go from like 0 to 100%. So again, tadalafil in this study came out to be the best choice for men with spinal cord injury. So changes in the expression and function of other PDE5 pathways. We have obstructed urinary bladder. Here's a study where we obstructed rats using a surgical partial ligature. They increased in size the prostate, the bladder. Here you can see that we're looking at bladder weight, which more than doubled. And we showed that we could co-localize PDE5Is in red, you see there, with alpha smooth muscle actin, which would be the smooth muscle filaments, and you get this nice overlapping on the right, negative and positive control. We also did that with humans, taking men with bladder outlet obstruction, comparing that to brain-dead cadaver men, and we saw a similar co-localization. So PDE5s induce cyclic GMP stabilization. That's the key thing. And in cardiovascular disease and cancer, you can see the PDE5 there. And when you look down the bottom, you're seeing it expand into so many other areas: anti-inflammatory, vascular control, cardiac regulation, anti-cancer. And my last couple slides are on PDE5Is in cancer. So cyclic nucleotides act as second messengers for promoting protein kinase activation, which in turn promotes apoptosis signaling. The cyclic GMP and cyclic AMP signaling pathways are aberrantly regulated in several human cancers, displaying different characteristics. Both cyclic AMP and cyclic GMP are shown to display anti-proliferative and pro-apoptotic effects, and in lines with these observations, altered expression of PDE isozymes have been reported in different cancer specimens. Also, I don't have time to go through it all, but in breast cancer, we have a lot of similar findings where the increased PDE5 expression is shown in clinical samples of breast cancer patients at both MNR and protein levels, and was correlated with tumor grade and stage. Prostate cancer, PDE5 inhibitors are also on the horizon and actually being used as they are in the bladder. So PDE5 inhibitors can sensitize cancer cells to chemotherapeutic agents. They can reduce extrusion of the chemotherapeutic agents by the ATP cassette transporters, ABCB1 and the others. And sildenafil and vardenafil treatment of breast cancer associated fibroblasts reduced chemokine expression, thus inhibiting cancer progression. And so I'm at Cooper Medical School, which is in Camden, New Jersey. We opened in 2012. I was one of the founding members of the school, and luckily we're still accredited, and we're moving along after 12 years. So thank you.